what's happening when more difficult things or more complicated things happens in the future. Especially that dynamics is not an easy topic and it needs some background on it. Let's start with the introduction. And we are just going to do a quick introduction. What is Volt? What is resistant state connectivity? And so to start, always like to start with this. That is one of the first experiments on discovering how we can get magnetic resonance signals, functional connectivity from the brain. And it was around the early 90s when it was discovered that there is a relationship between blood oxygenation and magnetic susceptibility. That a lot of experiments start going on how to use this information to get signals from the brain. And what we came up back then was to measure magnetic susceptibility as a proxy to estimate how much energy is spent in the neuron, actually in groups of neurons, from the capillarity in blood in the brain. So we basically estimate the amount of oxygen in the blood to see how much the cell is consuming or how much active is the cell at that point in the brain, and put that in fMRI images. And we take different shots on time. So it's a time-dependent modality where we know what's happening in the brain at certain points in time. And so what do we do with this? One of the first things that was done was to study different parts of the brain, and you find regions of interest. In this shot, you can see we chose the frontal lobe, some part in the frontal lobe. And you just take your region of interest and take an average of the activation on each of the scans that you get at different points in time. And of course, the activation change with time. So your activation will have a time course, it will have a time sequence with different activation values. Of course, we don't have to do this just for one part of the brain. We can take two different regions of interest and estimate the activation and make a correlation with them. And what this correlation tells us is how synchronized are they activation across time. We call this synchronization functional connectivity because we think we are activating at the same time if they are fully synchronized, and they might be performing a similar task, although we cannot call it task. Okay, so correlation can tell us a lot about how connected two parts of the brain are. However, we are not interested just in two parts of the brain. Usually we want to get a set of parts of the brain, a set of ROIs, and estimate how much are they connected among them. From those sets of ROIs can be selected by hand. There are atlases that you can use. In trends, we prefer to use line source separation, and in that line source separation, our preferred algorithm is group ICA, which is integrated in our software that allow us to find the regions of interest of more importance for certain brain illnesses. At the end, all we have is some brain location and how that brain location change with time. So we have a brain location and a time course telling us the changes in activation. And because these are many parts of the brain, it's difficult to just look at them as numbers. So we put them in a correlation matrix or correlation metrics, identifying the parts of the brain that are fully connected, more connected or less connected. It's just a nice way of visualizing. I'm going pretty fast. Visualizing the connectivity in the brain. Of course, there are parts of the brain that are connected in a positive correlation way and others that are in a negative correlation way. And this pattern is pretty common. It's been studied several times where the default mode network is expected to be anti-correlated with the rest of the brain, or at least most of the rest of the brain. And so, but it's not the unique correlation. The only correlation pattern you will see, however, is the most common. And if you take single value correlations, you will see it. So in here, I want to now start to define a little bit what is dynamic connectivity. Yes, sure, we can take two parts of the brain, two ROIs, and from those regions of interest, we can estimate a correlation value. For this particular example, we got 0.58, which is a good correlation value, quite high. However, you can see that in between the middle, there are more things happening than just a single correlation value. For example, if you take the first part of it, something between 1 and 3 seconds, you get a correlation value of 0.08, which is very low. If you take values in the middle of it, you get a correlation value of 0.95, which is extremely high. And if you go further in time, the correlation value drops again. So the 0.58 correlation value that you saw at the beginning doesn't reflect all that is happening in the brain activation and in the brain connectivity. In fact, in this example, we have 3 different correlation values, and they don't reflect what is happening on the whole time course, because there are only 3 points in time that we chose independently. What we really want to do is to track what is the correlation change along time. So instead of choosing just 3 points in time, we take a sliding window, which is that we take a piece of the information, estimate the correlation, and then move every time course, every TR of the scan, we move the window until we get a better history of what is happening with the correlation in time. These represent much better the connectivity and the connectivity changes that are happening with time than the single value correlation that we got if we just take all the time points and correlate all the time points. So, similar that we had in static connectivity, we don't have just 2 regions of interest. We have a set of regions of interest, and every region of interest has a different correlation history with time. So similar as before, we want to take the time courses and the correlations and estimate every time point. We want to estimate a correlation value and slide a window similar we did with the 2 regions of interest case. We want to use it for all of them. The only problem is in this case, we have a correlation matrix for every point in time, which we call it a window. It's a window in time that we use the term a lot. But it's basically that every time point has a correlation structure, and every time you move the window to a different point, you get a different correlation value. And that describes, in a sense, what is happening with dynamic connectivity on the whole brain. We, in studying dynamic connectivity, we find that there are persistent connectivity patterns. The matrices that change with time doesn't change randomly and don't change drastically. On the contrary, they have a smooth change with time. And some of the connectivity patterns that you will see will last for some time. In this example, which is just one part of the characteristics we see in dynamic connectivity, this is just an example. So I'm just exemplifying that, for example, state 3 lasted for 2 seconds before switching to a different connectivity pattern. And the next connectivity pattern lasted for 4 seconds before switching to a different connectivity pattern that lasted for 3 seconds. The example shows how you can estimate dwell time, which is how long in time a connectivity pattern is persistent. These connectivity patterns, we call it dynamic states. And the reason for calling a state is a little bit deeper than just the connectivity pattern, but we call it a state because they might represent some state of the brain. Since the connectivity could be representing what the brain is doing at a certain point in time. So these connectivity states last for a certain number of seconds before switching to a different one. And beyond just thinking that they are just connectivity patterns in the brain, they seem to have connections to what the brain is doing. For example, in this study, it was related that states of mindfulness where the default mode network is anti-correlated with the rest of the brain. Uh, those states of mindfulness seems to be connected with the anti-correlation of the default mode network. However, states of distractions or mind wandering is called in this study, where the brain is not concentrating in a particular thing. It's not doing mindfulness. We can see that the default mode network is not fully anti-correlated with the rest of the brain, mostly positively correlated. And they found out that this state is more connected to perception of mind wandering on the subjects that were not practicing mindfulness. So there might be a connection between the dynamic states we see in dynamic connectivity and the actual function that the brain is doing, the actual state in which the brain is doing. Of course, this is still under research and a lot there to do. But I think it illustrates a little bit the relationship between what the data is telling us that is happening, different states and time, and that there is a relationship between that and what the brain state actually is. So I'm just gonna do a couple of examples of the research that we want to pursue in the dynamic connectivity concentration. One is that we have discovered that some of these connectivity patterns are sequentially oscillating in time. This was found in a study, and I missed to put the reference. But you can see these are two connectivity patterns that are very similar, and the only difference is that they are of opposite sign. The positive correlation in one is negative on the other one, and vice versa. And if you look at the time progression, you can see that they go in sequence one after the other one, creating an oscillation that lasts for some time and then switch to a different type of oscillation or a different type of dynamic connectivity. But this oscillation that occurs give us evidence that there might be some attractor behavior in the brain, which is expected because the brain is a time-changing system. This is just a comparison between real data on two parts of the brain and the Lorentz attractor that is very well known in dynamic system theory, so that we have an idea that these attractors are happening in the brain, but they need more characterization because they're very high dimensional, and this is just 2 dimensions. So there's usually hundreds or thousands of dimensions in the data. We found out that those attractors can be disrupted by brain injuries. And this is a study that, well, for now it's in medRxiv, but it's under development to characterize well how the attractors are disrupted and recovered over time. The next one I would like to talk about is transcranial magnetic stimulation. And as you may know, transcranial magnetic stimulation is a way of non-invasive stimulating the human brain. So in dynamic connectivity, we found out that there might be a response on dynamic connectivity when we stimulate the brain. The oscillations we were explaining in the previous slides were found to be related to the stimulation in the frontal dorsolateral prefrontal cortex in the figure. And the last one I would like to talk about is creativity. We are studying also how dynamic connectivity is related or changes on different tasks related to creativity. For this study, we designed different creativity tasks. One of them was improvising, and I put Bart Simpson here just rapping, doing some improvisation, but the people were musicians who were improvising inside the MRI machine. The next task they were asked to do was to perform a piece of music they had previously memorized. And in this case, there is less creativity because they don't have to create anything. The thing's already created, and they have to retrieve from memory. So it's a different task. And well, during the experiment inside the MRI scanner, they performed each of this task in sequence. I forgot to say that some of the tasks were imaginative, they just imagining, and the other ones were actually performing the experiment. So we changed the task inside the machine, creating changes in time with connectivity. And these changes in time and connectivity resulted in differences in the connectivity, whole brain connectivity matrices. So we want to study more how different tasks can change the connectivity matrices, and this could help identifying if there is some illnesses in the brain, because a given task must create a given connectivity matrix, and if it doesn't, that could be an indication that the brain is not activating the way it should be. But that's for a future research. For this one, we just study creativity. And I just want to thank you for listening. I think I took a lot of time. So I pass here the screen to Robyn Miller.
Do you want questions now, Victor, or we can discuss after Robyn presents?
I can take some questions right now if you want.
If anybody has any questions about what Victor just presented, while Robyn gets her screen going. I think, Victor, you might have to unshare your screen for Robyn to share hers.
Oh, okay. There you go.
All right, cool.
Is this working?
Yeah. We can see the slide.
Okay.
It's not in presentation.
No, I understand it's not in presentation. I just wanted to see.
Yeah, we can see your screen and the slides. Yep.
Let's see.
Perfect. Looks good.
Okay, so I'm going to kind of extend from where Victor left off. I'll do a quick little rehash of the current kind of norms in studying, dynamic network connectivity at the whole brain level, and then move into some of the recent innovations that our group has been working on for a while, that hopefully might suggest some additional directions or I guess sub-directions that might be worth pursuing. So just shown here, what you'll see again later is a basically on the left, you see medial slices of one subject's brain advancing in time from left to right, and then diagonally, so it's just kind of a big 2D time series of one subject's brain. The one on the right is a different subject. And I will further discuss like what you're seeing there and why it's interesting soon. So, all right. Standard approaches. As Victor was saying, usually we decompose the brain into, we just kind of we map out the brain and kind of collapse the spatial particulars into nodes that then have just nodal identities. And then those nodes, as those nodes with indices or identities, have time series that represent the contribution of that spatial region to the overall signal at each point in time. And but at that point, we're studying, as Victor was pointing out, something more like correlations between the time series that represent the contributions of these parcels within the brain. And for a long time, this kind of study was done primarily on the full duration scan. So it was just, there was one such measure for each scan. You got one matrix, and it was somehow assumed that there's no evident canonical timescale, right, for this sort of thing. But people, whatever length their scan was, they just kind of used that length and reported the results. And that went on for a very long time. And then eventually, let's see, this is just more of that. We started just trying to capture some of the variations in this kind of measure of connectivity, which is correlation based. And so what we were doing, again, as Victor was indicating, is rather than using the entire time series, we will take the same measurement on shorter windows and slide them through the time series that you get one for each of these windows within the scan. So that creates like a kind of explosion of data, but it gives you potentially more information about what's happening in anyone's brain during the scan. So that the initial kind of pass at what to do with this gargantuan pile of high dimensional data was just to kind of throw it in a bag and cluster it. So, and this is still pretty normatively done. So after we do this, and each of these measurements is a time indexed measurement, it does have a window it belongs to. We sort of drop all that info and then just kind of pile them up, cluster them, find some cluster centroids, and consider those, kind of parameterize the process as a Markov model in which the states are represented by the centroids of the clusters. And we are going to study the process, each individual scan, as a process of transitioning between those states or occupying those states for certain periods of time. So it's basically, we're estimating a hidden Markov model from a whole pile of data and the centroids of the clusters that we get from that data. And so one of the newer directions now is to treat the dynamically reorganizing brain connectivity as a continuous process with contingencies, which is to say memory, and with maybe some energy constraints. So to dig in a little more deeply with this kind of measurement. So why are Markov models kind of bad for brain dynamics? There's some obvious reasons. So resting state fMRI, the connectome is very smoothly varying. It does not exhibit any abrupt phase transitions of the type you see in condensed matter physics. This fact arises from biological constraints and the constraints of the both imaging modality, which is very slow and based on vascular flow, and furthermore, additionally then from the smoothing effect of sliding window correlation. So it's a super smooth process. And another reason this might be limiting is that the brain supports behavioral and cognitive processes that are highly branched and contingent. And the Markov assumption would be particularly limiting for this kind of memory-laden dynamics. So, in the interest of time, I may just move forward from here, but to give an idea of what the shortcomings, let's say, of just kind of clustering the data can be, on the left, you'll see a plot that represents the ratio of the distance of each observation from the centroid of the cluster to which it was assigned over the distance of that same observation to the next closest cluster centroid, which is kind of asking the question, what does it really mean to belong to one of these clusters? Is it meaningful at all? And the mean of that ratio is 0.9 something or other. So the answer is sort of almost just as much in some other cluster. So it's just because it's a highly continuous high-dimensional process, and this is, kind of if you dig in, you see there are some issues there. And then on the right, there's just a schematic showing you have some sort of non-forgetful router state, and it's taking in kind of sequences of connectivity and can determine the next 3 frames, for example, of this connectivity movie based upon the entire sequence, not based upon just the most proximal, the Markovian assumption, where it would only the most immediately previous input would matter, since both of these inputs have the same immediate previous input to the non-forgetful router state. It's just a schematic idea. So let's see. This is a lot. So
this is the gummy worm slide, right?
This is gummy worms light. So one of the challenges, because the process is extremely continuous, extremely smooth, extremely smoothly varying, right? We want to be able to capture something more organic about the flows within the high dimensional state space, which is really hard. There's a lot of challenges because of the fact that we work with resting state data that does not have any, where across different subjects, time points don't really have specific meaning. And there are other challenges to doing this. And yet it seems like breaking out of the like kind of the discretization and the assumptions about hard state transitions that we have in the hidden Markov model approach might be fruitful, might get us somewhere. So in this particular approach to the problem, what we're going to do is take these long sequences of high dimensional dynamic connectivity observations. In this case, it is time aware, right? As a given sample, and the sequences are temporally, they're in correct temporal sequence. And we're going to take all of these sequences for all the subjects and try to find an embedding in a space that we can visualize, at least to start with, that preserves some of the key dynamic features, and from which we may be able to then extract sort of dominant directionality and patterns that we can then lift back up into the high dimensional space to construct something that is basically a movie that's important and representative, and also biologically plausible, that comes from a certain way of actually digging into the data through this lower dimensional embedding. And so the way this has gone, I won't torture you with the details. It's very cool because of the gumminess and colorful. And what we do is we're using an embedding algorithm called UMAP, Uniform Manifold Approximation and Projection, and This particular algorithm has two parameters, two main parameters, and adjusted properly, you can get an embedding of this extremely high dimensional space that, without any supervision whatsoever, does actually create the conditions, the qualitative conditions of the high dimensional dynamics in the low dimensional embedding. And so in these cases, you'll see individual subjects land along kind of continuous curves. The continuity within individual subjects is highly preserved. And another feature of the high dimensional dynamics at the group level is that it's super dense and entangled. So everyone is extremely close to observations from other subjects. And I want, for group purposes, I wanted to preserve that as well. And so with this parameter setting, it's a fairly loose range of parameters, but this was one that worked. We also have the entanglement feature, right, where everyone is within sample continuity and across sample kind of dense entanglement. And then we're going to work with that and try to find, there's a really cool slide that I didn't include, but what we're doing is we're constructing local linear approximations pointwise along all of the embedded curves, which is creating like a world of tangents kind of walking through this whole space. Really, really cool looking when you zoom in on it. And those are going to be then we're going to take those and try to find the kind of dominant directions at different points in the embedding and walk along those exemplar linearizations in 2D, averaging the embedded neighbors of each point in that linear exemplar in 2D, and then invert. So okay, let's see. The basic point would be, here's an exemplar, and here's a point on this exemplar. And near him, not everything is embedded. So what you need to do is find the 25, let's say, nearest embedded points, because the embedded points have inverses. And so you take the 25, let's say, nearest embedded points to some point on each exemplar, and take the mean of their inverse images in the high dimensional space. And then you're going to create a uniformly spaced set of points along each exemplar, go to the next one, do the same thing. Next one, do the same thing, until you get to the end of the exemplar and you get the final frame of the associated high dimensional kind of movie version of this summary tangent in the plane. So this is really different, and it's interesting. One of the things that's crucial to see is that the direction of the summary tangents is important. You can't just, so the direction does matter. And so you take two, for example, linear exemplars very proximal to each other in the embedding. So they would be grouped together. Individual observations along those curves would be considered very similar, but the curves are separate and the tangents are separate, and they end up clustering into two separate directions. And in one case, you can see some overlapping points right here in this oval and this oval. That is because the two line segments intersect, and that is where they intersect. But they are basically developing quite differently. They're getting to that point differently and exiting that point differently. And moreover, in one case, there's not much of a group difference. This kind of evolving pattern contributes equally to schizophrenia patients and to controls. This one, which kind of starts off in a hyperconnected condition and then develops forward from that, this one has a significantly larger role in reconstructing the sequences, the connectivity sequences of controls. And within the schizophrenia population, it also has a significant positive association with delusions. So it has symptom relationships within schizophrenia and also a group difference in its contribution. And these are basically things that kind of live in the same part of the embedding, but are pointing in different directions. All right, so another kind of fun thing that you can do with this approach, it's not quite there yet because we don't have enough observations, but you can look for sort of basins of attraction for the different groups in your study. So if I had enough healthies and enough patients, I could just use the embedded curves rather the tangents to the embedded curves for the controls in one world, and the tangents to the embedded curves for the patients in another, and then plop a point down in that embedding and have it walk along the kind of strongest gradients until it stops. And it will be different because these are different curves with different tangents. And what's interesting in this toy example, admittedly, is here we plop a random point in the same place in both embeddings, and they really go different places. So for the healthy guy, he's drawn all the way out to this kind of boundary area, and this looks like which actually represents a very structured kind of connectivity. The starting point for this in the schizophrenia embedding leads quite the opposite direction and takes him into something a little bit more messy. And if we were, for example, to ask what the basin of attraction, so this point X is clearly in the basin of attraction for this boundary curve. For schizophrenia patients, you can actually find a basin of attraction for the same boundary curve, but it's smaller. So, you have to be much closer to be sucked into that one. Anyway, this kind of thing there's somehow forward motion I see us being able to get from some further developments, along these lines. It's just something we haven't been able to do before. And wait a minute, what time is it?
It's 2:45.
Okay. Okay. So I'll just say a few more things. So another thing we can think about is incorporating sort of the energetic cost of certain of connectomic reorganization. We often don't do that. We just kind of measure like the distance. The distance traveled between two observations would just be, let's say, the pointwise L2 distance between them, without factoring in anything about the realism of or the plausibility or the energetic cost of that particular transformation of the connectome. So we're trying to work on this a little bit with optimal transport. So this is a little movie. So this is on the right is the graph representation or a graph representation, since it is isomorphic up to any sort of, is a complete weighted graph. That's true of all of our connectomes, right? They're complete weighted graphs. And so you can just like move all the dots around. It's still the same graph. And this is the typical presentation that we would look at if we were looking at it in a neuroimaging paper. It would have that kind of modular square look, but that's just for our convenience. And so you're watching some dynamics. You can see it on the left, you can see it on the right, everything, the edge weights are changing, right? This is sort of what's happening in the dynamic connectome at the graph level and in the matrix representation. And the idea here is that, okay, like leaving out all of the details that would make this more interesting.
We can have you back later, don't worry.
Yeah, so, is basically, so you take an observation such as the one in the middle, okay? And you can see that from a functional perspective, the observation on the left required highly globalized reorganization of function, like it's functionally integrated in a totally different way. There was some sort of widespread collaborative reorganization of the connectome to get to the image on the left. Whereas on the right, it was a simple kind of uniform decrementing of a very small amount. So they look very similar, they're highly correlated, but they're not identical. And there's a difference of like 0.01 between each of the cells, not visible necessarily to the naked eye. But the L2 distance between the middle and the right and the L2 distance between the middle and the left is identical. There's really no recognition of any type of potential energetic difference or higher level of difficulty or something in getting from middle to left versus from middle to right. And if you're using the optimal transport distance, you see something that makes a bit more sense here, where the distance between the middle and the right is considerably, orders of magnitude lower than the distance between the middle and the left. Seems like maybe some kind of progress there. Devil is always in the details, but that's, one direction we can start moving along. And yeah, so there are some differences in key measures of dynamics. If you look at the dynamics measured with L2 versus measured with the OT distance, the direction of certain vector versus.
Robyn, can we close off?
Yeah, we can totally close. Let me just I'll zip through these slides and I won't say a word, just like cool pictures, and then we'll wrap.
Let's hold the slides. We'll have you present those again at a later point. Don't worry about it.
Okay. It's getting just to the cool stuff.
You couldn't help yourself. Okay.
No.
I want to give us. So we have 10 minutes left, and I want to just move to discussion. If you want to just one or two closing words, feel free.
All right. Nope. No closing words. Just.
Okay. All right. Thank you, Victor, and thank you, Robyn. Sorry to kind of cut you off there, but I did want to make sure we end this, at an hour, and that we have at least a little bit of time for discussion. So, what if folks, have any questions about the dynamics? I guess, there's obviously the mathematical, aspects of this, and then there's also kind of why is it useful? What can we do with it? What can we study with it? Kinds of questions, which hopefully different people will have thoughts on. So I'll just open it up and see if folks have any questions. Feel free to just jump right in.
I actually have a question. Can you hear
me? Robyn, unshare your screen so we can see.
All right. I'm actually here.
Yay!
I'm having some issues with unsharing. Give me a sec.
No,
I had a question because of the question of the robustness and the reliability of the measurements. We know that in neuroimaging, our sort of retest replication is not what you would call fantastic. Mind you, I can do the same thing with my blood pressure, to get retest replication issues. But I was wondering how all of these dynamic measurements are affected are robust to differences in the imaging data protocol, are robust to times of day, are robust to retest reliability, all those kinds of questions. Has any of this been, I know we've applied it a lot in sort of case control differences looking at the aggregate, but at the individual level, how informative is, if I scan you today and come up with all of the worms, for your brain, how much will that constrain what I would expect to see tomorrow if I scanned you again?
So especially on let's say the worms, I think that there are two points. One is part of what's happening with the worms is group level, right? So we are trying to so you'd have to scan everybody again, and then we're getting these linear exemplars based upon an entire group presentation of these trajectories. And I think in that context we get a lot of the same trajectories or dominant directions, but it may be that different subjects are in different parts of it. I think that the group level would be more robust than the individual level under conditions of, scanning on different days under different, because in a way, I guess my general idea, you want it to be robust to scanning parameters and up to a limit, some kind of perturbation in scanning parameters. But it may be, robustness can be important or can be a sense, it can be an indication of dullness of the measure, right? Because if the person really is different, they're more animated, they're more tired, they're more stressed. If our measure doesn't find that, then it's a dim-witted measure. And so but we don't ever know exactly what we're, when we scan someone 4 or 5 times over the course of a month, we see differences. We don't really know if it's reflecting the things we would want this measure to reflect, or if it's just a mess as a measurement, not, finding like the variability within the person.
And if I can comment, separate from so it also depends on the method, right, that you're using to estimate. And we've looked at this in a few different approaches. There's one study that Anissa Brule did, which was using so it's not the high dimensional approach Robyn was mentioning, but it was using the sliding window approach. And that showed, quite high replicability. And in particular, the metrics for dynamics were more robust than the static measures were. So it may well be that when you're dealing with like resting fMRI data, given that people kind of are doing different things over time, if you characterize those and compare apples to apples a little bit better, that you would improve your ability to do that. But then the caveat is, of course, and everybody, I think a lot of papers get this wrong. When you're looking at dynamic measures in somebody's brain, you don't necessarily want to. It's not necessarily the right answer to say we get the same pattern every time, because people are in different conditions, right? And so I think that's a really tricky problem to address. But we can look at sort of how robust it is. Is it something we see consistently when we look at large samples? Is it robust to things like, motion, for example? And I think it is, in many cases it is. But.
Yeah, I also wanted to comment a little bit on this. That's a problem that we have been thinking about for a long time. And as you said, it depends on the mood and what the state of the individual is.
Literally.
So, and it's probably impossible to correct fully. It's very, very random. So instead of that, I think one of the things that we were trying to do, especially with stimulation, TMS or TES, is just make the brain get into in a predictable state. So if we could, for example, stimulate the brain and bring the connectivity to a state we know the brain is going to go to, so we can replicate the same state every time. So that's one of the areas where I think DMAP could try to do good research on, trying to get the brain into a predictable state and then estimate brain illnesses from that state. That goes along with the data we were seeing in attractors, where brain injury just changed the attractor. It's because it changes the connectivity behavior. But so the question is, how do we get the brain to a certain position, certain connectivity, so then we can predict from them? And well, another possibility is do something such as mindfulness. So get the person to relax, to non-stress them as much as they can. But it's more difficult because some people can, some others cannot relax easily. So TMS or TES seems to be actually a stronger and less non-invasive. We're still depositing energy there. It's a little bit more strong way of doing it, but it could be part of the answer we are looking for.
Other questions? Comments, thoughts? Does this intersect with any of the work that folks that aren't necessarily doing this might find interesting? I guess, are there some common points we could explore? Or is this just kind of crazy out there stuff? We still have Andre on. Andre, I'll pick on you.
Pick on me. What do you mean? It's
Where
a
do you
it's
start
a
it?
it's a very interesting question. Even in our business, it depends on the application, right? And again,
Hmm.
things like structural stability or robustness, again, I'm talking about the question that originally was asked. You have to redefine for certain systems, right? They don't apply directly to typical dynamical systems. Okay, when we say something that is robust, again, it's more a physical term, right? When we talk about in terms again stability, we have to define what kind of stability. There are different kinds of stabilities depending on applications, right? So which means you can't just use that term loosely, right? You get to be more careful. Right. And then structural stability, sometimes I like it. I mean, in our applications, I like it even more because it basically means you don't really rely on the parameter variations. I don't care about parameters anymore, right? I'm interested in having a pretty flat kind of region, so it doesn't depend on. I mean, again, I don't know if Sergey is here or not, right? It doesn't depend on the changes in a I don't want to say parameters because again it's also confusing whether they're intrinsic parameters or they're external parameters. So it's a touchy issue. We have to sit down and then come to the language, right, that we all agree upon, right, and then pursue from now on. Because again, let me just say that the terms and notions from straight coming from dynamical systems often are not applicable to applications under consideration. So it's a trick,
Yeah.
right? And then Vince, I did want to come to the room, and just an unfortunate chain of events. I'm here on campus.
Yeah.
And then we have this march, and then they block the streets here. So right before the meeting.
I'm having trouble still getting used to walking between spaces versus just virtually jumping, right?
The police was everywhere, just like a 1:45.
Yeah.
Right? They blocked the streets.
Yeah. All right. Well, oh, sorry.
I
Yeah,
don't have anything.
go ahead, Eddie.
I don't have anything specific. I just wanted to introduce myself. I'm Eddie Namias from the philosophy department. I don't know how often I'll get to join y'all but I will say if anybody is doing this sort of work specifically on looking at resting state potential or what the brain people are doing.
Uh-oh.
We lost you, Eddie.
I'll turn it. Huh?
Can you repeat it? We lost you for a sec.
Oh, sorry. I was just going to say I'm particularly interested in what the brain is doing when people are imagining things or just during resting state potential or doing counterfactual reasoning or thinking about alternatives. I'm interested in free will. So if anybody's using these methods for any of those techniques, I'm always happy to hear about it. Thanks for inviting me. I'll try to.
That's great. Yeah, thank you. Hopefully, I'm sure there's some interesting things that we can explore there. So yeah, it'd be fun to.
Well, we certainly have some data sets on that.
Yeah, definitely lots of mind wandering. Yeah, the free will one is obviously an interesting one. So, but thanks. Well, it's being mindful of time. It's 3:01, so I will let everyone go. But thanks again. Next week we're going to do a talk on reading, right?
Yep.
So
No imaging, just reading.
Yeah, just reading, which is dynamic too.
Yep. Y'all are gonna tell me how it works.
Absolutely. So, so thanks everyone for joining, and have a great. I guess it's Friday, right? So have a great weekend too. Okay. Yeah, Thursday's great. Jess and Kyle and everybody, you can hang on. We'll have our little chat in this room. So. I was gonna see if there's Dina, if there was a way to oh, since it's recorded, we might have that. But can we like keep track of the participants or the number of participants or something in some way? I don't know that we need too much detail on that, but just sort of like, we had like 31 people today.